Plasma levels of the thyroid hormone Triiodothyronine and Thyroxine are maintained within a narrow range. Too much can lead to hyperthyroidism, and too little can lead to hypothyroidism. So, how are plasma levels of T3 and T4 kept within a narrow range? Plasma levels of T3 and T4 are maintained by the thyroid-stimulating hormone, or TSH. TSH is a glycoprotein with an alpha and beta chain. The alpha chain is identical to other glycoproteins like luteinizing hormone and follicle-stimulating hormone. However, the beta chain has a unique structure that provides binding specificity to the TSH receptor. Also, you need to know that TSH is glycosylated, which means that it is bound to sugar residues like glucose. These sugar residues are thought to regulate receptor binding, activity, and clearance of TSH. Now, TSH controls the synthesis and release of T3 and T4 by regulating most aspects of thyroid hormone production and release. It starts when TSH binds the TSH receptor, which is a member of the G protein-coupled receptor family. Binding of TSH to its receptor activates the G-alpha protein, which then activates adenylate cyclase. This leads to the production of cyclic AMP. It is the increase in cyclic AMP that promotes the following. First, it increases iodide uptake by the sodium iodide Symporter. Second, it increases iodination of thyroglobulins. Third, it increases the conjugation of thyroglobulins. Fourth, it increases endocytosis of iodinated thyroglobulins. Fifth, it increases proteolysis of iodinated thyroglobulins. And sixth, it increases secretion of T3 and T4. It also promotes increased proliferation of follicular cells, which helps increase T3 and T4 production even further. This is called hyperplasia. So, how is plasma TSH regulated? Well, TSH synthesis and release are positively regulated by thyrotropin-releasing hormone, or TRH, and negatively regulated by T3. To start with, TSH is synthesized and released from specialized cells called thyrotropes, which are located in the anterior pituitary. This process begins when the thyrotropin-releasing hormone, or TRH, binds the TRH receptor, which belongs to the G protein-coupled receptor family. Binding of TRH to the TRH receptor activates the enzyme phospholipase C, or just PLC, which then leads to the formation of diacylglycerol, or DAG, and inositol triphosphate, or IP3 for short. DAG activates protein kinase C, which leads to the phosphorylation of downstream proteins and enzymes. These proteins and enzymes in turn stimulate the synthesis of new TSH, while IP3 promotes the release of calcium ions from internal stores, which leads to the release of vesicles by exocytosis. To keep TSH levels within a normal range, T3 inhibits the synthesis of TSH within thyrotrophs. This is referred to as the negative feedback loop. It begins as T3 and T4 are transported into the thyrotrophs by the monocarboxylate transporter. Following uptake, T4 is immediately converted into T3 by the enzyme type 2 deiodinase. As the concentration of intracellular T3 increases, it binds the thyroid hormone receptor, which has been shown to interact with other transcription factors like ADA2 and Pitt1 to inhibit the transcription of TSH alpha and beta chains. The T3 negative feedback loop onto the thyrotrophs is one of the primary mechanisms by which plasma T3 and T4 levels are maintained within a narrow range. So TRH stimulates the synthesis and release of TSH, while T3 inhibits the synthesis of TSH. In the next lesson, we'll talk about how TRH is regulated.